This module is about catalysis, and specifically about enzymes. Key properties of enzymes include that most are proteins, although, as you may know, several of them are actually RNA molecules, and those RNA enzymes are called ribozymes. We'll talk about those later in the course. There are about 3,000 different enzymes per cell. That means that cells will have overlapping sets of enzymes. In other words, one cell won't have 3,000 enzymes and another cell has 3,000 totally different ones. They're simply overlapping sets of enzymes, and these characterize the different cells with their different functions. Like any catalyst, enzymes accelerate chemical reactions. How do catalysts work? Well, here's a reaction occurring very slowly in the absence of a catalyst. Reactants are converted to products slowly. Versus a catalyzed reaction, in this case catalyzed by an enzymatic catalyst, happens very fast. And when we look at enzymes, we're talking about substrates being converted to products. So we use the word substrates for reactants in enzyme catalyzed reactions. Let's compare enzymes, biological catalysts, with some inorganic catalysts. Inorganic catalysts are manifold, but they include many of the rare metals, nickel, platinum, silver, and so on. Even hydrogen gas is a catalyst. And typical enzymes have names like pepsin and trypsin. These are digestive enzymes originally discovered in the stomach in the case of pepsin, in the pancreas, actually in the small intestine, but coming from the pancreas in the case of trypsin. Those are old names. More recently, meaning in the last 30 to 50 years, we've named most enzymes with the ASE at the end. So we have ATP synthase, which does like it says, it catalyzes ATP synthesis. Ribonuclease catalyzes RNA breakdown, which would then mean that deoxyribonuclease catalyzes DNA breakdown. We'll learn common names like these as we go along and look at different enzymes. Again, all enzymes and catalysts have in common that they increase reaction rates. In both cases, the catalysts, enzymes or inorganic, are unchanged at the end of the reaction. Inorganic catalysts are generally nonspecific, so shavings of platinum can catalyze a large number of different chemical reactions. Enzymes tend to be highly specific. Most of them actually catalyze one and only one reaction involving only one substrate or set of substrates and cannot really catalyze any other chemical reaction. Inorganic catalysts are rigid and inflexible, as you would expect from metals. Enzymes, however, as you would expect from any protein, are flexible, and they can undergo shape change or allosteric change. Allosteric just means shape change or alternate shape. Inorganic catalysts cannot be regulated. This is largely because they are rigid and inflexible. Enzymes can be regulated, and this is in fact due to the fact that enzymes can undergo allosteric change, that is, change in shape, and we talk about the regulation of enzymes as, for the most part, allosteric regulation. Let's look at the mechanism of enzyme action. We want to look at two things: structural considerations. These are the shape of the enzyme, and remember that can change, and the affinities of enzymes for substrates. We also will look at energy considerations, and we'll talk about free energy and activation energy. So here's a picture of an enzyme and a substrate. The substrate's the little red structure at the upper left. The specificity between the two of these things, the affinity, the attractive power between the two, brings the substrate, which in this case is glucose, close to the active site of the enzyme, and the enzyme in this case is hexokinase. There you have the ASE suffix right at the end of the name. A hexokinase we will see in looking at glycolysis, so it's not a strange enzyme. In fact, it's a very common enzyme, the first one of glycolysis. So a mutual affinity draws the two of them together, and then the substrate penetrates the active site, achieves a better fit of substrate to enzyme, and that's called induced fit. This sort of phenomenon has been actually identified and seen by X-ray crystallography, in which you can look at the shape of the enzyme in the presence or in the absence of substrate and see that their shapes are actually different. The place where the substrate bound is called the active site of the enzyme. The induced fit mechanism predicts intermediates in the interaction of substrate with enzyme to form product. So we have it drawn here as enzyme plus substrate being drawn to one another. When they are in fact bound to one another, we could say that there is an intermediate we can call ES star. This is an initial intermediate. Then comes induction of fit, where the substrate somehow fits even more perfectly into the active site of the enzyme, and that complex is then called the enzyme-substrate complex. In this form, bonds in the substrate or substrates are being strained and stressed in preparation for actually rearranging them to do the chemical reaction. Finally, we have an enzyme and its products, and the products have lower affinity for the enzyme, and so they dissociate from the enzyme. You could postulate a third intermediate called EP, an EP complex. We believe the EP complex for most enzymes is very short-lived, in fact, because of the very low affinity of products for the enzyme, and so that we don't assume it lasts very long. But for reasons I can show you later, we do assume that the enzyme-substrate complex has a finite lifetime, albeit very short. It's still finite and needs to be considered in understanding how an enzyme works. So it says, understanding the interactions of substrate or substrates, if there's more than one, and an enzyme are essential to understanding enzyme kinetics. Now let's look at the energy side of the mechanism of enzyme action and talk about activation energy. On this graph, I have plotted free energy versus time, and the graph that just appeared is a graph of the progress of the formation of C and D from A and B in the reaction shown. A plus B goes to C plus D. If you look at the chemical equation for this reaction, you will see that it is talking about an exergonic reaction. Why? Because the equilibrium arrows have the longer arrow pointing towards the product. So what's happened here? Let's go back and do this one more time. In the progress of this reaction, there's a moment where there's a large input of free energy in order to get the reaction to go, in order to form something called the transition complex. In this instance, we can call it an AB complex. So A and B are brought together so they can interact at the surface of this enzyme. That's an energy barrier. Why is it an energy barrier? Well, if you think about it for a moment, if you put A and B in a test tube or in the cytoplasm of a cell, molecules of A and molecules of B will tend to diffuse away from one another, won't they? They'll, just by natural diffusion, they won't stay together. They're not attracted to one another. The function of an enzyme, however, is to provide a surface, called the active site, which will draw A to it and B to it by virtue of mutual affinities. And by drawing A and B to the same site, the enzyme is in fact overcoming entropy, right? It's overcoming the tendency of the molecules to exist in a disordered state. Well, this energy barrier can be overcome by enzymes, but we'll see also this activation energy barrier can be overcome in several other ways. We can lower the activation energy barrier simply by raising the temperature in the absence of any catalyst. This is not done in living things, of course, because cells don't regulate their temperatures as a rule, certainly not enough to make enzymes act considerably faster or slower. We can throw in an inorganic catalyst in a test tube for a chemical reaction that might occur in a cell. So we could conduct that reaction at standard temperatures at 298 degrees Kelvin in the presence of an inorganic catalyst, and that would even more efficiently lower the energy barrier to the progress of the reaction. But the most efficient thing to do is to use the cell's natural enzyme to convert A and B to C and D. That is the most efficient way to lower the entropic barrier to this reaction. As I pointed out a moment ago, this reaction is exergonic, and you can see that because I've bracketed the free energy that is released as A and B form C and D. You might like to go home and with a piece of paper on a pencil, this is not an assignment, but to help you understand it, draw the same reaction in reverse and look at the activation energy under those conditions if the reaction was in fact endergonic instead of exergonic. So how do enzymes lower activation energy? Well, as I think I said, their surfaces, their active sites, have a high affinity for the substrates, for A and B in this example, and they bring the substrates together. They don't merely bring them together, they bring them together in the right orientation to allow bond rearrangements. So again, activation energy is the free energy needed to overcome entropy. Well, enzymes don't act in isolation. Biological catalysts are not in the cell isolated from other reactions. Typically, they're part of biochemical pathways, such as the one shown here, where A is converted to B by enzyme 1, B to C by enzyme 2, C to E by enzyme 3, and so on. As molecules of E build up, it's possible that one of them might actually go back and bind to enzyme 1, changing its shape and preventing it from acting. That's called feedback inhibition, which is a very common way of regulating enzymes to control the levels of in this case, levels of E in a cell. So when E builds up, when the final product of this pathway builds up, very characteristically, some of those molecules go back and bind to an enzyme early in the pathway, changing its shape, thus inhibiting it. That's called feedback inhibition. It is a reversible phenomenon, so that when levels of E drop because the cell is using it for some purpose, the E molecule that is bound to the enzyme 1 would actually come off, and then the pathway could resume at full speed. So we control enzymes by, in fact, controlling their shape, by allosteric regulation, as I indicated earlier. So here we have the same enzyme and the same substrate, and we have an allosteric effector, which in this case is going to bind to the enzyme at a site other than the active site, but it's going to cause a change in shape at the active site, which prevents the substrate from even attaching to the enzyme. So the inactive enzyme is no longer able to bind the substrate. And that brings us to the end of this presentation.